What I'd like to do is discuss the impact that two advances have made on medicine. One is systems thinking, and the second is big data and its analytics. And these have led to this idea I'm going to talk about now of scientific wellness, and we've explored it in some detail. But what that gives us is an incredibly powerful big data platform for exploring both wellness and disease and looking at it, both of them in dimensions that has not before been possible at all. And I'll start by talking about a series of paradigm changes I've been involved with since I started at Caltech in 1970 that kind of inexorably have led to this idea of systems medicine and a healthcare that's predictive, preventive, personalized, and participatory. We call that P4 medicine or P4 healthcare. But the first of these changes was to develop a series of instruments for reading and writing DNA. And what they did was brought to biology for the first time, one, the ability to generate large amounts of data very, very quickly, high throughput data for biology, and two, the idea of big data and its analytics and what you can do with it. And one of these instruments, the automated DNA sequencer, got me involved in the first meeting ever held on the Human Genome Project. And I was fascinated with that because in beginning to think about how you dealt with the complexities of both biology and disease, it was really clear that we had to do what later came to be called systems thinking. And doing the genome was the key to beginning about thinking of systems thinking in humans because it gave us a complete parts list of all genes and by inference all proteins. And curiously enough, when we developed the automated DNA sequencer amidst enormous skepticism when we started, we found that it required bringing together engineering and chemistry and computer science and biology. And that led to the idea of the imperative necessity for having a good cross-disciplinary framework on which to operate biology. And of course, the idea was leading-edge biology should always be able to drive the development of relevant technologies, and they in turn create a demand for the appropriate analytic tools. And if you could in one place put all of those things together, you really could transform biology and come to all sorts of new hypotheses about what it was. And I tried to do this at Caltech and failed for interesting sociologic reasons, but Bill Gates made it possible to do it at the University of Washington. And for 8 years, that first cross-disciplinary department was enormously successful. But what I really wanted to do was place on top of that then a center, an institute for systems biology. And it became clear that the easiest way to do that was to resign from the university and start the institute as an independent nonprofit research institute. And of course, systems biology, which we'll talk about in some detail, is a global, holistic approach to both biology and disease, and it's global and holistic both in the sense of providing the technologies and strategies you need, and it's global and holistic in the sense of putting together the big science that's necessary to solve most really difficult problems. And we'll talk a little bit more about that in just a moment. Immediately when we started the Institute in 2000, we focused on disease, and that very quickly led to these concepts of systems medicine and P4 medicine. And so in about 2013, 14, it was clear we'd like to figure out how to bring these new approaches plus big data to the healthcare system. And in 2014, we did an experiment I'll talk about a little bit later on scientific wellness that embodied the absolute essence of P4 medicine. And we started to show it could work on ordinary individuals very, very effectively. And of course, the final event in this series of paradigm changes was in April of last year when the Institute became the research arm of Providence and I, the chief science officer. So that opened up the ability to bring these efforts focused around systems medicine and big data directly to the patient's bedside. And we'll talk about how we're going to be doing that toward the end of this talk. Now, the Systems Biology Institute, I think, really was an unusual organization. It focused immediately on strategies for systems thinking, both for biology and disease. It focused immediately on the development of technologies that were necessary to carry out systems biology. And it spent an enormous amount of time developing complementary computational tools for doing that. And of course, it created this cross-disciplinary platform that let biology drive technology, drive computation. And we've found that as we look at most biological problems, they really are big science problems. They can't be solved by a single lab with a limited number of skills, but rather you have to put together strategic partnerships and bring complementary talents together and focus them on really hard and challenging problems. And I'll show you examples of at least one of these just a little bit later on. And of course, what has really turned out to be important too is this whole idea that science is really about creating knowledge, and it is imperative that the knowledge that's useful for society be given to society in some ways. And we found the best way to do that is to create spin-off companies that use new kinds of biology. So the Institute, for example, in 16 years now has created 8 companies, and some of them are going to be very big, successful companies, and we'll talk about one of those in just a few moments. So what exactly did systems thinking bring to systems medicine, or P4 medicine? It brought 2 things. First, it brought this incredible, powerful new platform, which we call dense dynamic personal data clouds, where for each individual you generate enormous amounts of data of many different types. And you end up with clouds literally that have billions of data points. And the idea is you can integrate and analyze these data so that you can reveal for each individual the actionable possibilities that, if acted upon, will let them optimize wellness and or avoid disease. And we'll give you some examples of that in just a few moments. And what I really want to express is that these dense dynamic personal data clouds are not only powerful for understanding wellness, but they're really going to be transformational for understanding disease. And we're just starting to look at them in this context. So this is exactly what precision medicine should be. Precision medicine as practiced by NIH today is largely genomics, and I think accordingly is incredibly short-sighted. The second contribution to systems medicine has been this idea that biological networks are the informational byways of regulating development and physiologic responses, aging. And when these biological networks become disease-perturbed, they cause disease. And if you can identify the difference between a disease-perturbed network and its normal counterpart, you can begin to understand the basic pathophysiology of the disease and get candidates as biomarkers or targets for drug therapy. And I'll show you some examples of those a little later on. You know, the other point I'll make is you should never use the word pathway, because that implies a two-dimensionality that is very limited. All networks are three-dimensional interactive networks. And the reason it's easy to get around any one block in the network is you have many alternative pathways to follow. And that's conceptually a really important framework for realizing that in cancer, a single drug is never going to give you a cure, except maybe in very rare and exceptional cases. Cancer is going to have to be treated just exactly like AIDS, starting with multiple drugs. And how many probably will differ depending on what kind of cancer you have in mind. So about 2013, well, 2007 or 8, we had really clearly defined systems medicine and P4 medicine. And we were thinking about how we could begin to bring them to the healthcare system. But we realized we had a whole series of technologies that needed to be developed. And we found it extremely difficult to get NIH and federal funding because of the conservative nature of proof you can do it before we're going to give you a grant to do it. So we were really fortunate, I was really fortunate in 2007 to meet the Minister of Finance for the state of Luxembourg, who wanted to bring biotech and healthcare to Luxembourg. He asked us to write a proposal to help them do so, and we did a lot of things for them I won't talk about, but what they did for us was to give us $100 million over a period of 10 years to develop a series of technologies and systems-driven strategies we'll look at in just a moment. But what these did was to place us then at an incredible tipping point in 2013 where we could really begin to start bringing P4 medicine to the healthcare system. So some of the technologies that we've been engaged in following, and I'm not going to talk about any except to say there is a third-generation DNA sequencing technology being developed that is single-molecule analysis, nanopore, nanochannel-based, and electronic detection. And my prediction is within a 5 to 8 year period, when this technology comes to the fore, it will make human genomes $100 a piece, and you'll be able to do a human genome in 15 minutes or so. And that'll change completely how we view genomes. Now they cost $1,200 or $1,300 a piece or so. The strategies that we developed included family genome sequencing, included new approaches to diagnostics. And in fact, I'll go through and give you a very high-level glimpse of some of the things that we've really looked at. And the first is obviously relevant to the brain, looking at a neurologic disease induced by activated prion particles in inbred strains of mice. And the importance of this study is you could look at the onset of the disease and follow it all the way through to the termination. You could follow the entire course of the disease. And what was interesting, we looked at brain transcriptomes, and we were able to identify about 300 genes that probably encoded this neurodegenerative response and so forth. And we were quickly, through histopathology, able to show there were 4 major mechanisms that were employed in this neurodegenerative process. And about 200 of the genes mapped into the networks associated with each of those 4 things. And the other 100 defined another 6 networks that completed the whole neurodegenerative process. And when you looked at the dynamics of the networks, you could explain virtually every aspect of the pathophysiology of the disease. But the really important point is we could detect at the very beginning the earliest transition when the prion replication accumulation network first became disease perturbed. And that's important because we're going to show in the future how important it is to identify the earliest stages of any disease. And the important point to realize in that, we identify the earliest stage of the disease about 13 weeks or so before there's any phenotypic manifestation of the disease. So you see the disease really before it manifests itself. And I'm going to show you later, we can do exactly the same things for humans in interesting ways. But one of the questions is, if you wanted to think about doing this in humans, you couldn't obviously do brain assays. So what we developed and spent an enormous effort on is identifying proteins in the blood that could be recognized by targeted mass spectrometry that were brain specific. And what that meant is if you had a normal brain, the 100 proteins we recognized had one set of concentrations they expressed. If the brain became disease-perturbed, those proteins that were a part of the disease-perturbed network would change their concentrations. And since each disease uses different combinations of disease-perturbed networks, you could distinguish health from disease, and if disease, which disease it was. And in fact, what we were able to do is actually go back to the mice and take their blood and use 11 brain-specific blood proteins that mapped into each of these 4 networks and show in the blood exactly the same sequential pattern of disease perturbation of the 4 networks, offset by 10 days or so from when we saw it in the brain. So the important point is you can, in the blood, follow the initiation and progression and ultimately response to therapy with these brain-specific blood proteins. I think they're going to be an enormously powerful tool for studying brain diseases, cancer as well in the future. A second area that we've really spent a lot of time pioneering is showing that if you take a family that has a disease, multiple affecteds with the disease, by sequencing the entire genomes of the family, you can identify the disease gene just from a single family study. And we've used this very powerfully to show for many simple Mendelian traits, we can identify the disease genes. But what we did more recently was to study 41 families with bipolar disease, a complex multigenic disease. And what we demonstrated is, one, that these 41 families had 125 genes that were mutated, that were associated with the disease, that all of these genes fell into one of 2 functional categories. They were related to GABA receptors or they were related to calcium ion transport. So they made a lot of sense in terms of the brain biology. And number 3, we went to 2,000 sporadic individuals and showed that the 25 top gene mutations were also present in these sporadically chosen individuals with bipolar disease. So this gives us the tools for thinking about really complex brain diseases, schizophrenia, autism, as well as bipolar and other kinds of diseases. Now, we've developed a whole series of really powerful strategies for using mass spectrometry to compare normal bloods and disease bloods to get biomarkers that actually can be used clinically. And I won't go through and describe the many strategies we use to separate signal from noise, because that's the big challenge. But I will say that we've been very successful in getting diagnostic panel for brain cancer that has the ability to distinguish benign from neoplastic nodules. And in doing so, you can save the healthcare system $4 billion a year. And that's the reason 4 major payers have adopted this test to supplement their analyses of lung cancer. Number 2, we've used a similar strategy in preterm birth. And again, have a 2-feature panel that at 19 weeks can unerringly predict who's likely to get preterm birth, and one then, for example, can use progesterone to delay pregnancy and avoid the preterm birth. And then we've done a series of things that are more in the discovery phase. They're not as far along, but they include markers that can distinguish soldiers from Afghanistan that have severe PTSD, post-traumatic stress disorder, from their normal counterparts. And we've actually done recently Charles and collaborators a study of glioblastoma and have 4 really interesting proteins that look to be good predictive markers and so forth. And we've done the same with organ-specific proteins for liver and been able to look at fibrosis and toxicity and a number of things like that. And the final approach that I'll take may be one of the most revolutionary that you're going to see in this study, and that is the real limitations with drug discovery today are, one, making the libraries is really hard and complicated and expensive, and number 2, screening them is very, very expensive. So what Mike Tyers, a colleague in Montreal, has done is devise a really incredible new technique where the big idea is, one, you literally can make billions of synthetic natural gene products. And number 2, you can use the high throughput tools of biology to screen them. And in doing so, the screens will cost 1/10 to 100th of penny per screen as compared with analyses ordinarily in drug companies of $2 or $3 per screen. So it's far more rapid, it's far more complete. The limitations in the natural chemistry creation of drugs is you only search a very small dimension of drug shape space. With this approach, you can search an enormous dimension. And the idea is microbes have an enormous number of metabolic enzymes. DNA synthesis is very inexpensive. So you can put 4 or 5 or 6 enzymes together in blocks that can be expressed coordinately in yeast to make compounds that are the assembly of what the 4 or 5 enzymes can actually do, creating natural synthetic products. And you can screen the individual yeast either against molecules or against cells to get the desirable targets and so forth. And Mike has actually demonstrated this works beautifully by synthesizing 1,600 of these genes and putting them together in a combinatorial fashion in a series of these blocks. And we're hoping to persuade him to move to Seattle and set up a big drug discovery entity that will be associated with a lot of the clinical practices that we're talking about here. So in 2014, there was this really interesting convergence of systems medicine and digital medicine and big data and its analytics and social networks and so forth that let us sharply define what P4 medicine is. And I just want to say it really differs from contemporary medicine in striking ways. It's proactive. It's focused on individuals and not on populations. It looks at wellness as well as disease, not just disease. And it's all about using this platform, the dense, dynamic, personalized clouds, to study wellness or disease. And what that platform allows us to do for the first time is to assess in the individual, in the context of the individual, the relative contributions of genetics and of environment. And genomics alone is obviously one dimension. Yeah, I wonder if you could put some shades down up here, is one-dimensional. But the other thing it's all about is saying that the way we discover drugs now, where we put 20,000 patients in a pool and give them drug or placebo, and then abstract the results, is all wrong, because every one of those individuals is unique genetically and unique environmentally, and you can't average different things without enormously increasing the noise to signal, and drug companies have demonstrated that absolutely adequately. So for P4 medicine, we would take 20,000 data clouds, each individual with their own data, and we would stratify based on that individual data. For example, patients into groups of responders or non-responders to drugs. And just to emphasize the power of that, let me say these are the top 10 selling drugs in the US today. And the colored individual is a responder, and the blue individuals are the non-responders. So in these populations, you have anywhere from 1 in 4 responding to 1 in 25 responding. So the vast majority of people, in some cases, pay enormous amounts for drugs, and they get all the complications associated with the drugs. And with the classic screening techniques, you have no way of distinguishing responders from non-responders. But if you use the dense dynamic data clouds, you automatically get the differentiating features and so forth. So I would say P4 is about wellness on the one hand, it's about disease on the other hand, and I'd make the argument that we're going to create a whole new sector in the healthcare industry, which we call scientific wellness, and I'll define that for you in just a moment. And I'll argue that in a 10-year, 15-year period, the market cap for scientific wellness will far exceed that for the healthcare industry. As you know, the 98% of the dollars for healthcare spent on disease now shift quite rightly over toward wellness. And of course, what is really important to realize are the uses that you can apply these dense dynamic data clouds to. So one, and I'll show you this in just a moment, we can use it to optimize wellness. Number two, we can actually use these data clouds to follow individuals over time if you have large populations, and we will very soon, and follow wellness to disease transitions. We can get biomarkers to look at the transitions, and we can identify the drugs that could reverse early disease before it ever manifests itself as phenotype back to normal. So this will be the preventive medicine of the 21st century. And three, of course, we can use these clouds to look at disease themselves, to stratify them, to follow the progression of the disease, the response to therapy, and if fortunate, the return to health and so forth. And I'll just say, N of 1 experiments in the future are really going to be important for deconvoluting a lot of the complexity associated with many contemporary diseases. So in 2014, I persuaded 108 of my friends to submit for a year to this procedure of dense dynamic data clouds to study wellness and see if it was really going to make a difference. And of course, the really key thing, of course, is that we were able to use the data clouds to assess both genetic and environmental contributions. And the data that we generated was complete genome sequence. And then every 3 months, we took blood, urine, and saliva for clinical chemistries and analyzing proteins and metabolites. Every 3 months, we took fecal samples to get the gut microbiome. And then we used the Fitbit to get activity, quality of sleep, blood pressure, a whole variety of other kinds of things. And the idea then was that we would spread out then the analysis of these data, which, as I said before, led for each individual to actionable possibilities to optimize wellness or avoid disease. And every month we had coaches that called the individuals and presented them with the actionable possibilities in the context of what their own health objectives were. And I'll say these coaches are utterly critical. That human touch meant that 70% of the actionable possibilities were actually acted upon, which is really a remarkable number. The data that came from these things was really quite striking. And I've likened it to the Hubble telescope, which looked at the heavens with a resolution heretofore unachievable. So too these dense personalized data clouds were able to look at human biology and disease at a resolution that wasn't possible before. And from the study, this year-long study of these 108 individuals, we were able to identify many, many striking correlations that gave us new insights into disease mechanisms, biomarkers, drug targets, and things like that. We were able to show that you could actually use GWAS data in the context of your whole genome sequence to estimate your relative probability of getting any one of about 60 different diseases, including Alzheimer's and many different kinds of cancer and so forth. And that relative disease estimate is really useful for looking at individuals in clinical trials in the future. And I think that's going to be an important part of things. And then finally, we were able to use the data clouds to see wellness to greater wellness transitions, or wellness to disease, or disease back to wellness. So all of these things provided powerful new insights into these things. So I think this platform is really going to transform both wellness and disease. And to give you a specific example of the correlations, we were able to identify about 4,000 correlations that tied together one bit of data in each of the 6 different data types with other bits of data in the other types. And the statistical correlation revealed all sorts of insights into systems that were interacting with one another, that were interrelated to one another, that became perturbed in disease. It gave us insights in how do you stratify individuals with particular diseases. But to give you an idea, if you took all 4,000 correlations, they looked like that hairball on the left. And you could take a very simple algorithm and just ask it to clip the edges that represented the lowest probability associations. And you quickly saw beautiful structure of subdomains of related kinds of proteins. And it turned out those subdomains actually all represented functional domains in the physiology or the disease of individuals. And from those, you could get really interesting biomarkers related to particular processes you'd never thought before, or even drug targets. And I won't talk any more about that. Now, the 108 individuals, I will say, half of them went in skeptical, and at the end of this year, I think all of them felt that it had been a really significant life experience. And some of the conclusions they drew are that it was absolutely incredible that with knowledge you could actually determine your own health course. And I think this is actually going to be a key to bringing down the cost of healthcare. It was clear that genetics didn't determine your destiny, rather just your potential. In many ways, you can get around a lot of genetic limitations and so forth. But what was really interesting is most of the 105 weren't nearly as well or healthy as they thought they were. And in fact, if you thought about wellness as a big staircase, I would say the average person is very close to the bottom. And of course, what brings you up various steps of this staircase are these actionable possibilities. And of course, the interesting thing is, as we get more data, we enormously expand the repertoire of actual possibilities. So in a sense, we're going to indefinitely keep expanding the possibility for making yourself well. And of course, the real idea is to elevate one up this ladder to Eric Topol's group of people called the Welderly, 90-year-olds that are mentally and physically in perfect shape and functional and active, never had a drug, never been in the hospital, kind of ideal individuals. And the question is, through executing on actionable possibilities, can we bring ordinary people up? And I think the answer is we can bring them a long, long ways, but it makes this scientific wellness a lifelong vision. And of course, at the end of this thing, then what virtually all the pioneers came and said, what's going to happen next? So we started a company called Arivale, which is a company that brings scientific wellness to consumers. And it now, after a little more than a year and a half, has about 1,700 individuals that are associated in this program. And we've just started to spread it down to Washington and so forth. But what's really been interesting is after we've learned how to do scientific wellness, we really figure by 2020, we can actually scale it to where there are 1 million individuals followed long term. And this means we're going to see transitions of wellness to disease for virtually every kind of disease that one has to worry about in the future. The affiliation with Providence, which made ISB the research arm, for us was very exciting because Providence is a big entity, 7 states, 50 hospitals, 100,000 employees, sees 5 million patients a year or so. And they, interestingly enough, have more than 30 million electronic medical records, so enormous opportunities there for data mining and everything. And so in terms of being their research arm, what we decided to do was to create a series of translational pillars. And we've listed the first 7 that we're looking at here. And we took common ordinary diseases for the most part, and we're going to focus, 1, on using these novel systems approaches that others have not used and systems-driven technologies. And number 2, we're going to focus for all in using this platform of dense dynamic data clouds to follow initiation, progression, response to therapy, and so forth for these diseases. And I'll just mention a little bit more about 2 of them. So for Alzheimer's, we're collaborating with a UCLA partner who's taken a really interesting systems approach to optimizing how you can maximize effective synaptic communications. And in doing so, he's come up with a 36-point regimen that includes drugs and exercise and diet and supplements and a whole mental exercises. But he's had absolutely a remarkable success with 124 patients in the earliest stage of Alzheimer's, being able to reverse them back more toward normal. So we're going to set that up with 2 other clinical trials in Alzheimer's that are systems-driven in different ways to see whether we can, in fact, in a 3 to 5 year period, develop an algorithm for inevitably reversing at least early stage Alzheimer's. And the final area that we're going to focus on, and Charles mentioned some of the things we would be doing together, is Alzheimer's, is glioblastoma. And there we're really interested in a variety of things. One, I'm a big advocate of saying we should, in cancer, be giving people triple drug therapy from the very beginning, including immunotherapy. So we're going to start thinking about how we can analyze triple drugs and deal with the problems of effectiveness and combined toxicities and all of those other kinds of things. Immunotherapy in glioblastoma is challenging because glioblastoma is a low mutational load tumor. So you have to figure out ways to jack up the effectiveness of immunotherapy and so forth. But those are things that we'll do in the context of the environment here at Swedish and Providence St. Joseph's. So, the future of healthcare, we really have brought P4 medicine to the healthcare system through wellness studies that we're going to be doing with Providence. 2,000 of its employees will go into the Arivale program over 3 years. With proper controls, we'll be able to assess both the quality of healthcare and how it's improved and how it's reduced the cost of healthcare. We've applied this dense dynamic personal data cloud platform to both wellness and disease, and lets us assess both the environment and genetics. We've shown you can optimize wellness for individuals. We've suggested that when you follow lots of patients long-term, we'll get the earliest transitions and reverse them before the disease manifests, and that'll be preventing disease of the 21st century and so forth. We've suggested this scientific wellness is really going to be a lifelong objective. And of course, we're going to, I think, create a whole new sector in the healthcare industry of scientific wellness. I believe the data are absolutely going to transform how healthcare industries actually carry out their tasks. And that includes drug companies, nutrition companies, diagnostic companies, and the like. And finally, I'm going to talk in just a moment about how I think we can really bring scientific wellness to the poor as well as the rich and lead to a democratization of healthcare that we couldn't have imagined even before. And I think that context is really interesting. And so let me just say a few words about that. Today, it costs about $3,500 per year per person for scientific wellness. And in 10 years, I think we can get that down to 100. One, because we'll have done enough discovery so we can go from millions of tests to perhaps 10,000, decrease in 10 to the fifth amount of information we need. Number two, I think we'll have a device, a cell phone-like device that can take a drop of blood and make all of these measurements and ship the data out for the kind of analysis that we've talked about here. And I think three, one of the questions is, how do you scale coaching? And I'm really intrigued by the use of avatars that are instructed by artificial intelligence. And I think they're going to be able to do marvelous things. Whether they really can do everything the coaches can or not, I'm not convinced, but it's really worth thinking about. And of course, we're going to then be able to use the wellness, the more effective ways of dealing with disease, the ability to reverse things at their earliest stages to make arguments that are, I think, really going to be compelling to the payers so that insurance will be available in a 10-year period. And I think the amount of savings we're going to have, and this is a low estimate, is 5 times the amount it costs to take care of an individual patient per year, per day. And we could run through why I think about all of that. And of course, I think in the future, healthcare will be generally available through the government or not, so that the poor as well as the rich can be reached, and we can reach out to all of those. And on my last slide, I've just listed some of the things I've talked about here that I think really are going to have a big impact on brain research. So optimizing wellness, particularly brain wellness with these blood-specific proteins we can assay. Being able to detect early and stratify diseases and generate biomarkers for them, and ultimately drugs that can deal with them. Using family genomics to identify relevant genes in the complex disease processes and so forth. And of course, using the synthetic natural product approach to generate billions of drugs that can be screened to look at new targets very, very rapidly. And this is the approach that's going to let us deal with drug resistance really effectively in the future. The Gates Foundation is really, really interested in that application. So, and I think single-cell analyses are going to allow us to ravel complexity at a whole new level we've never, ever thought before. But anyway, I think the future is bright. There's a lot to do, and I hope you have a terrific meeting the rest of the day.